Hello, and welcome to my poster. My name is Sarah Wright, and I'm a bioinformatics PhD student at UCSD. And I'm excited to tell you about our work, State of the Interactomes, an evaluation of molecular networks for generating biological insights. So as I'm sure you can appreciate, the amount of data we've generated about genes, proteins, and the relationships between them has been growing exponentially over the last few decades. While this has powered countless discoveries, it also leads to the challenge of how do we actually represent and analyze all of this knowledge. Biological networks are interactomes, where each node in the network represents a gene or protein, and each edge in the network represents known relationships between those genes, are a powerful way of tackling this complexity. However, anyone wishing to embark on a network biology journey faces the obstacle of choosing from among the dozens and dozens of available interactomes. In this study, we comprehensively evaluate 44 publicly available interactomes on common network biology tasks, showing that in some scenarios, bigger is better, while in others, more specific is better. So first, let me introduce the interactomes we're evaluating. In figure 1 here, I'm showing a meta-network of interactomes. So here, each node represents a publicly available interactome, with the color indicating the construction method, the experimentally derived, expert curated, or a composite of other resources. The size of these interactomes varies from 10,000 interactions to over 10 million, and the arrows show that there are extensive dependencies between the different sources, with central databases such as BioGrid and Intact being the most frequently incorporated into other interactomes. To assess the interactome quality, we first looked at disease gene prioritization. So in general, we expect that genes implicated in the same diseases will be involved in similar biological processes and therefore closely linked within an interactome. So we defined sets of disease genes from DisGenNet, purple, and the GWAS catalog in yellow, subsampled each gene set, and assessed the accuracy of recovering the held out genes using an area under the precision recall curve metric following network propagation. The results showed that a few of the large composite networks, such as String and HumanNet, consistently have the highest performance. In addition to the raw results in the first and third columns, I'm also showing size relative results in the second and fourth columns. These reveal that some of the curated networks, such as DIP and HPRD, have some of the best performance on a per interaction basis. So even though these networks are smaller, the information contained in them is of high quality. Given that the large composite networks tend to have the best performance, we then asked whether there are certain types of information that drive this result. To test this, we defined evidence type specific interactomes from HumanNet and GeneMania and reran the disease gene prioritization. We found that over both sources, domain similarity, pathway information, and physical interactions produced high-quality predictions compared to other evidences such as co-expression. Of course, disease gene prioritization is not the only application of network biology. As an orthogonal measure of quality, we also benchmark performance on interaction prediction. The motivation for this is that because our knowledge of biology is incomplete, our interactomes are also incomplete. However, we can use the knowledge that we already have to predict the most likely additional interactions and therefore accelerate our knowledge generation. To test the utility of each of the interactomes for this, we tested their ability to recover held out self edges as well as gold standard quorum complex and panther pathway interactions using a path of length 3 edge prediction algorithm. We observed that a different set of networks was highest performing on this task compared to disease gene prioritization. And the results for the gold standards show that, for example, in the second column, that experimental PPI networks are best for predicting quorum complex edges, which makes sense because complexes are made up of interacting proteins, and that the curated pathway and signaling networks tended to be best at predicting panther pathway edges. Overall, this means that matching the network to the task is important for interaction prediction. To summarize, network biology is a powerful tool for addressing the complexity of biological processes and disease genomics. However, the choice of interactome can be crucial for success. We've shown that a small number of large, composite networks, such as HumanNet and String, have the highest performance in prioritizing disease genes, and that physical interactions, domain similarities, and pathway relationships are crucial evidence types in building accurate representations of biology. Importantly, though, when it comes to different tasks, the optimal choice can change, with the specificity of the interactome being critical for accuracy when it comes to things like interaction prediction. In addition to the results presented here today, we've also examined how well each network represents the breadth of biological processes and are investigating the trade-offs involved in customizing networks to specific disease contexts. If you're interested in learning more, please stop by my poster on Wednesday or feel free to reach out to me via the conference platform. Thank you for stopping by.